Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. And in this context, in the current module, we are discussing about the different ways in which you can be able to measure the enzyme activity. So if you recall, in the previous lecture, we have discussed about the different aspects and how on different types of assays what you can actually be able to set up to measure the enzyme activity. Now, when you talk about the enzyme activity, you are actually going to talk about the several types of enzyme catalyzed reactions. These enzyme catalyzed reaction could belong to the catabolic reactions or the anabolic reactions. So anabolic reactions are the reactions which are going to be used for synthesizing the new molecule, whereas the catabolic reactions are the reactions which are where the biomolecule is going to be broken down or oxidized to generate the energy. Now, when we talk about the enzyme activity, this means we are actually going to talk about that the enzyme is actually processing the substrate, and in that process the enzyme is actually going to generate the product. Okay, and if you want to measure the enzyme activity, and if you recall in our previous lecture we have discussed what are different ways in which you can be able to express the enzyme activity. So one of the classical way of expressing the enzyme activity is that you are going to express the enzyme activity in the units, right? And every unit is actually going to be defined as the amount of enzyme required to convert a 1 micromolar of substrate into the product in a given time or 1 minute. And this we can calculate. So enzyme activity can be calculated if we want to, if we can be able to measure the consumption of substrate or the production of the product, right? Now, if I want to measure the substrate or the product, I could actually be able to use the exclusive property associated with this particular substrate or exclusive properties associated with the product, right? So in that case, so in many cases, we can actually be able to see the disappearance of the substrate. In some cases, you can be able to see the appearance of the product, right? When you see the appearance of the product, it is actually going to give you the positive signal. Okay, so this is actually going to give you the positive signal, which means you are actually going to see a increase in some value, right? So it is actually going to give you a positive signal. Whereas when you are going to see a disappearance of the substrate, then it is actually going to give you the negative signal, right? What is mean by the negative signal is that your starting molecule, for example, if you started with 100 molecules of substrate, then after, so at time 0, for example, right, at time 0 you have started with 100 molecules of substrate, right? After 10 minutes it is actually going to be 80, right? So suppose 100 molecules of and suppose we are measuring the absorbance of this particular substrate, right? And this is actually exclusive, right? So and does not have the any kind of cross reactivity from the product or the enzyme. Then, for example, if you have the absorbance of this 100 molecule as zero point 0.8, for example, then 80 molecule will actually going to show you an absorbance of 0.6. After 20 minutes, you are going to have an absorbance of 60, 40, like that, and ultimately it is going to be 0, right? So similarly, you can actually be going to have a decrease in absorbance, right? This means you are actually going to know what is the amount of substrate going to be consumed if you are actually, so you started with 100 molecules, now you have 80 molecules, right? So you can actually be able to take this value and you will say, okay, zero point 0.2 OD substrate is being consumed in 10 minutes, and that is the way you can be able to use these values to calculate the enzyme activity. Similarly, when you are going to talk about the positive signal, you are actually going to say that same way, right? You can have the time, you can actually be able to say at the starting you are going to have 0 molecules, and you are going to have 0 absorbance. So it's actually going to say absorbance, number of product molecules, right? And like that. Okay, so time 0, you're going to start, there will be no product form, right? Because the enzyme has just started the reactions, and then absorbance is also going to be 0, right? Then after 10 minutes, you are actually going to see 20 molecules, right? This is not exactly what is happening, right? 20 molecules are going to be consumed, right? So 20 molecules are going to be produced, right? So 20 molecules are going to show you an OD of suppose 0.35, okay? Similarly, you can have 20, 30 like that, and it is actually going to show you 30 molecules, 40 molecules like that. So it's going to show you an absorbance of 0.5, and it's going to show you an absorbance of 0.7 or something. So this is actually the direct measurement, right? Here you are actually going to plot this. So 0.35 is the concentration of the substrate product, right? And you can actually be able to say that this is amount of the product is being formed, and that's why this is the same amount of substrate is being consumed, so that you can actually be able to use for calculating the enzyme. So that's why you see when you are measuring the product, it is easy and straightforward, whereas when you are measuring the substrate, you are actually going to go through with the another function, right? You have to always calculate the delta absorbance, right? You have to calculate how much absorbance is gone down, right? So in this case, 0.8 to 0.6, then for example, here from here to here, again 0.2, from here to here, it's going to be 0.1 like that. So here you have additional step, and when you have the additional step, more steps means you are actually going to increase the number of chances of incorporating the errors. And that's why it is advisable to go with the positive signal rather than the negative signal, or I will say you should not measure the disappearance of the substrate, rather than you can actually be able to measure the appearance of the product. So depending on the different types of properties of the substrate or the product, the enzyme assays can be done by the utilizing the different types of techniques. What are these techniques? You can actually be able to use the photometric assays. So you can actually be able to use the photometric assays, where you can actually be able to change in absorbance, or you can actually be able to change in the fluorescence, and you can actually be able to say luminescence and all that. And then you can also have the radiometric assays. So these radiometric assays, where you are going to see the change in the radioactivity of the substrate or the product. And then you can also have the chromatographic techniques, which you can also use for measuring the enzyme assays. And here you can actually be able to capture capture the substrate or the product, right? And you can actually be able to measure that with the help of either the radiometric assays or the photometric assays. And then you can also have the gel electrophoresis. So you can actually be able to visualize the product onto the electrophoretic gel, and that you can also be able to use for measuring the enzyme activity or enzyme assays. So enzyme assay is a very complicated recipe, or I will say enzyme assay is a complicated protocol, where you are going to use not only one type of assay. You may have the possibility of using the multiple combinations of different types of techniques. For example, you can actually be able to use the chromatography techniques to isolate the product, right? And you can actually be able to use this product, and you can actually be able to quantitate this product with the help of either the photometric assays or radiometric assays. Same way, you can actually be able to use the electrophoretic assays or electrophoretic system to visualize the substrate, and then you can actually be able to use either the photometric assays or radiometric assays to quantitate the product, right? So let's start first with the photometric assays, and we'll discuss different aspects of photometric assays, how you can be able to, what are the different precautions you should take when you are setting up the photometric assays, and when we'll discuss about the UV visible spectroscopy and as well as the fluorescence spectroscopy, how you can be able to exploit these techniques for setting up a fluorometric assays and photometric assays, and how you can be able to utilize that for measuring the enzyme activity. So photometric assays, as the name suggests, photometric assays means you are actually going to absorb, or you are actually going to see something with related to light, right? Photometric means, photo means light, metric means measuring, right? So you are actually going to do the light measurements, whether the light is going to be absorbed by the substrate, right, or product, or it is actually going to be that the substrate or the product is actually going to produce the light, right? Sometimes you can actually have to, we'll be able to see that if there is a product which is formed, it is actually, giving the light, right? Or sometimes you can actually have the alternative, right, where you are going to excite the molecules, and then it is actually going to give you the fluorescence, right? So fluorescence is also a phenomena where you are actually going to measure the light intensity only. Okay, so let's discuss and start discussing about the photometric assays. So as the name suggests, the photometric assays are the most commonly used type of enzyme assays. They are convenient and capable of producing the accurate and reproducible result on large number of samples in a short period of time. So photometric assays are the most popular and most important, and it is easy to perform, and in terms of infrastructure, in terms of instruments, in terms of the reagents, they all are very much cheap actually, so it's economically very viable. The chemical transformations that accompany the enzyme catalyzed conversion of substrate to product frequently results in a useful change in the optical property of the system under the investigation. So either there will be a change in the substrate absorbance, or there will be a change in the product absorbance. Okay, so either you can have a very exclusive product which actually can give you the exclusive the light absorption phenomena, or you can actually have a substrate which is going to show your disappearance. When the reaction is catalyzed by the enzyme under assay do not produce a useful change in optical property, the addition of the appropriate additional reagent frequently allow the reaction to be photometrically monitored. So these are actually going to be a couple of examples where neither the substrate nor the product are exclusive. Then you can actually be able to add some coloring agent, and that coloring agent is going to either react with substrate or the product, and then ultimately it is actually going to give you the colored change actually, and that's you can actually be able to monitor with the help of the photometric systems. The other photometric methods are less commonly used than enzyme assay based on change in light absorbed by the solution as the reaction processes. Change in the fluorescence and the turbidity of the solution, on the other hand, provide the foundations for useful optical method for assaying the enzyme. So before we discuss and understand the phenomena of the photometric assays and how you can be able to design the assays, let's discuss some basics of the photometry and how it actually works. So first thing what we have to discuss is we have to discuss about the absorption phenomena. So what you know is this is actually the UV visible spectra, right? And what you see is this is the radiation of the different wavelengths, right? So you started from the radio waves. You can actually be able to talk about microwaves. Then you have infrareds, and on this side you have the ultraviolet, X-ray, and gamma, right? So as you go from this side to this side, there will be decrease in wavelength, and that's how there will be an increase in intensity. And this is the region which is called as the visible light, or this is going to start from the from somewhere around 400 nanometer to 700 nanometer, right? So that is will be the called as the UV visible spectroscopy, and next to the 400 nanometer, it's go up to the 250 nanometer, or sometime it's going to be 190 nanometer, and that is going to be called as UV region. Okay, so UV region also can be used for photometric assays. So you can actually have the UV visible range of the spectrum, and that can be used for the all the photometric set assays. So visible light is of wavelength between the 400 to 750 nanometer. Compounds that absorb light in this range are colored. Many colorless compounds also absorb light in the UV range, that is from the 200 to 400 nanometers. And light absorption occur when the electron in a irradiated absorbing molecules are promoted to a high energy level, because the frequency of the electronic oscillation in the molecule coincide with the frequency of the irradiating lights. The wavelength of this frequency is that at which compound absorb maximum amount of light is depend on the structure of that particular molecule. The amount of light absorbed depend on the probability that the electronic collision occurs. And if you recall in our in a couple of module back when we were discussing about the the different types of UV visible spectroscopy, we discussed about that when we were saying that how you can be able to use the spectroscopy uh, for measuring the enzyme substrate interaction, right? That time we discussed about how the absorption is changed even if when you have a slight modification of one group, or there is a substitution of the few more groups, right? So remember that when we were talking about how the absorption phenomena is changing when the benzene is getting benzene is getting substituted at different unit values and so on. So second thing what we have to discuss is about the absorbance. So absorbance is A is equal to minus log I by I0, and where I0 is the light transmitted by the solution not containing the chromophore, right? So it is I0 is the light transmitted by the solution not containing the chromophore, which means that this is, and the I is the light transmitted by the solution that does contain the chromophore, okay? And A is the absorbance. So A is actually a ratio of I by I0, and because this is a ratio, it does not have any unit, because you, so that is very important, right? Many times even in the different types of exams, they will actually going to ask what will be the unit of absorbance. So it is a unitless property. How you can convert the absorbance to the concentrations, right? So absorbance is directly proportional to the concentration, right? So if you recall from the Beers-Lambert law, what Beers-Lambert law is says that absorbance is directly proportional to the concentration, and absorbance is also directly proportional to the path length, which means if you combine these two, absorbance is going to be equal to the epsilon CL, right? And epsilon is called as the molar absorption coefficient, and the unit is mole per centimeter, right? So if you keep the path length constant, right? If you keep the path length constant, then the absorbance, then the concentration is going to be the absorbance divided by the epsilon, okay? So when the path length of the cuvette or path length of the light path is 1 centimeter, it is going to be constant, right? So if it is a constant event, we can actually be able to ignore this, right? And that's how the amount of, as you will increase the concentration, there will be an increase in absorbance, and you can actually be able to calculate the concentration of the molecule with the help of the absorbance divided by epsilon, okay? This means if you can have the molar absorption coefficient of the molecule, you can take the defined, you can take the molecules, put it into the cuvette, and then if you measure the values, right? If you measure the absorbance, and then you can actually be able to just divide that value by the molar absorption coefficient, it will actually going to tell you what will be the concentration in terms of moles, right? And that is the very very powerful tool, right, to measure, right? There are deviations from the Beer-Lambert law, and that we are not going to discuss here because there are saturations, and then there will be a product dimerizations, and defragmentations and all that. So that is actually going to change the this particular relationship, but that is called as the deviation from the Beers-Lambert law, and that you can actually be able to study from any of the classical biochemistry book that, or biophysics books, like I think the, if you go through with the book Wilson and Golding, right? So if you think about this, right, you can actually be able to know this, right? So you can actually be able to study about the deviation from the Beers-Lambert law, and that is going to be helpful to understand under what conditions the law will not be applicable, and then we should be little more careful. Okay. Now let's talk about the instrumentation part. So instrumentation of the spectrophotometer. So all photometric instruments operated by the same basic principles. Light from a light source of a specific wavelength is passed to the cuvette containing solution of our interest, right, molecule, for which you want to measure the absorbance, which is then detected by the photometric detector, right? So what you see here is you are going to have a light source. Then in front of this light source, you are going to have the monochromators. So monochromator is nothing but a prism, right? So it's going to have a prism. And you know that when the light goes through the prism, it actually diffract, and that's how it is actually going to show you the different wavelength, right? And remember that when the white light goes into the uh, prism, it actually going to split into different 7 color, right? And since it got fragmented into 7 color, you can actually be able to rotate this prism, and that's how you can be able to select the wavelength of your choice. So same is here. You can actually be able to rotate this, and that's how your wavelength of your choice will enter into this particular slit, right? The slit will be a small gap between or the hole, and from there you can actually be able to allow the entry of this particular array, right? And then there will be a place where you can actually be able to keep the cuvette, and that's how the cuvette is actually going to have the molecule, right, for which you want to measure the absorbance, and that's how uh, uh, the unabsorbed light will actually going to pass through the cuvette, and it is actually going to be detected by the detector. And detector could be of many types. It could be photodiodes or photomultipliers. The wavelength of the absorption for different chromophore is different, right? And therefore, it is essential to restrict the wavelength of light source before the chromophore is exposed to it. Now, we will discuss about the different components. So, first component is the light source, right? The selection of the light source is predominantly depend on the wavelength in which the measurement are to be made. So, either the UV visible or UV range or the visible range. The most used light source for the glass enveloped filament tube bulb, the quartz enveloped tungsten halogen bulb, or the deuterium lamp, and more recently the xenon lamp. So many of these lamps are available. You can actually use the tungsten lamp, or you can use the halogen lamp, you can use the deuterium lamp, and you can also use the xenon lamp. And all they are actually capable of providing the light of the different wavelengths, so that you can actually be able to use according to your choice. And sometimes what happen is that in some of these spectrophotometer, you may have the two lamps rather than single lamp, right? So you can have the lamp for the visible range, you can have the lamp for the UV range. To cover the useful UV visible range, that is from 200 to 700 nanometer, an instrument using both deuterium and the quartz halogen lamp is usually necessary, although diode array instrument rely on a single. So low pressure deuterium lamp that emits sufficient light over the whole UV visible range. Okay, so you have the choice. You can actually be able to use the two lamps. You can actually be able to use the single lamp. Then we have the wavelength selector. So this is called as a diffraction gratings, right, or the prisms, right. So the reliability of the absorbance values will highly depend on the accuracy of the wavelength to which the chromophore is exposed. That is the specific wavelength, right. So selection of the lambda is very important, right. And that's why the wavelength selector is a very important component of the spectrophotometer. The colorimetric simply use a series of filters to filter out all the wavelength other than that of a required specific wavelength. But the availability of range filter is very small, and sometimes the wavelength to which the chromophore is exposed is a well away from its normal lambda max values. Okay, so there are many ways in which you can be able to use the wavelength selectors. You can have the filters, you can have the diffraction gratings, you can have the prisms, right. So depending on the price of the instrument, you can actually have the different types of things, right. and that's how you can actually be able to have the precision in terms of selecting the particular wavelength. And if you are going to have multiple wavelengths, it's actually going to, it's actually going to affect the final absorbance phenomena. The modern spectrophotometer uses a holographic diffraction grating to disperse the light from the source into a spectrum. Rotation of the diffraction grating varies the light wavelength of the element emergent light passing through a slit, and to extent to which this approximate to a monochromatic light depend on the wavelength of the slit, and it is described in the specification of the instrument as the bandwidth. So bandwidth is the deviation from the selected wavelength. Okay. Then you have the determination of the absorption spectra. So many photometric enzyme assay problems can be solved more quickly if the absorption spectra of a solution involved can be determined rapidly. This can only be accomplished in an instrument that uses physical movement of the diffraction grating to irradiate the sample with the monochromatic lights. This means you can actually be able to absorb, you can actually be able to have the absorption spectra of the molecule, right? So you can actually have like lambda on this side and absorbance on this side, and that's how you can actually be able to know where this particular molecule is absorbing, and that's how you can actually be able to choose this, and this wavelength which where there have the maximum absorbance can is called as the lambda max. So if you have that kind of instrument where the diffraction grating can very precisely be able to move around from the different positions, and that's how it can actually be able to illuminate the sample with the monochromatic light of different wavelengths, then it can actually be able to give you very precise value for the lambda max, and that's how it actually can help you to decide what lambda max I should use for the detection purpose. Modern instruments allow a full spectrum to be determined in less than 3 seconds. A diode array instruments capture all of the points on the spectra at the same time, and a spectra can be measured at 1 second intervals. One major drawback of this is that since sample is exposed to all possible spectrum, problems may arise. Okay. Then we have the cuvette holders. So maintaining the constant temperature is essential for the continuous assay. Electrical heating is preferred over circulatory water bath. This means you can actually have the the electrical heating, right, rather than water circulation, because water circulation, the fluctuation is going to be more. Many instruments have multiple cuvette holders, allowing the multiple continuous assay to be performed at the same time via the automatic mechanical movement of the carriage between the measurements. It is also ideal in having a cuvette holder which accepts cells of the path length other than the 1 centimeter. And the very important thing is the limits and the source of errors. So when you are doing the enzyme assays, you always have to be very careful about this particular phenomena, that you are going to have limits until you are, so you are actually going to have a range in which you are actually going to be utilized, or you can be able to use the assay. And then what are the different sources through which you are actually going to get the error, because if you know the sources, you can actually be able to minimize, or you can be able to take it into account so that you can be able to correct for those errors. One is the non-linearity arising from the stray light. Okay, so the light that originates from the instrument and passes directly to the photodetector without even passing through the cuvette is the source of this error. Remember that when you are actually having a bulb, right, and from the bulb you have a filter, you have a prism, right, then you have a slit, and then you have a cuvette, right. So if you have the solution, right, so there are actually going to be, so it's actually going to enter, right, and there will be some rays which are actually going to go directly, and they will actually going to hit the detector. Okay, and these direct light which goes to the detector are actually going to provide a source of this error. The deviation of the linearity between the concentration and absorbance is predicted by the Beers-Lambert law. It often due to the instrument rather than the complex behavior of the chromophore. Despite the best effort, this error will always occur, and it is accounted by the passing the light to the empty cuvette with the solid opaque object in the and nothing the absorbance. Okay, that means how you can be able to correct this? What you can do is just put this cuvette which does not contain any sample. Okay, so if that happens, there will be some amount of light which is actually going to be passed through without this sample is going to absorb, and that you can actually be able to measure. So imagine that if that light is IS, so if IS is the amount of light which is actually directly be getting into the detector, then you should subtract that amount into that light you should actually be able to add onto the intensity. So what you are going to do is your apparent absorbance would be that minus log I plus IS divided by I0 plus IS. Okay, and that is actually going to somehow or some way actually going to correct the error what is going to be incorporated from the instrument. Then we have the instrumental noise. So this is random fluctuation in the photodetector output that originate within the instrument, and it is this is and it's not caused by the solution. Okay. Then we have the zero drift. So in a single beam instrument, this is slow, steady change in the apparent absorbance. It is caused by the time dependent change in the photodetector and a lamp, which causes the true value of I0 to change, whereas the I0 value used to calculate absorbance is at that measurement when the absorbance 0 was set. Okay. So this is the problem of single beam instrument, right? When the single beam instrument, what happen is that when you have two different types of spectrophotometer, you have the single beam instruments, you can have the double beam instruments. So if you have a double beam instrument, you can continuously be able to monitor what is the intensity coming from the bulb and what is the intensity the detector is detecting. But if you have a single beam instrument, what will happen is that initially you put the cuvette and you measure the absorbance having the no chromophore, right? And that value you are always subtracting from the your measured value. But that value which you have measured 10 minutes back may actually change over the course of time because the bulb is actually going to get warm up, or there are some other artifacts which are actually going to happen. So those things cannot be mapped in a single instrument, right? Single beam instruments. And because of that, the double beam instruments are more preferred because they are actually going to give be more helpful. So when using a diode array spectrophotometer, this problem can be avoided by the internal referencing or subtracting the absorbance reading taken at a wavelength when there is no absorbance change due to the reaction. This problem is also avoided by using a split beam instrument that determines the absorbance from the I and I0 value measured simultaneously. Then in the photometric assays, you can have the continuous assay or you can have the discontinuous assay. So when you are setting up the continuous assay, which means you are actually going to have the enzyme, you will add the substrate, and then you are actually going to have the generation of product, and that you can actually be able to measure. So either you measure the substrate or the product. The continuous assay in which the enzyme catalyzed reaction is monitored as it occurred is preferred to one in which the enzyme reaction is run for a fixed time and then stopped before measuring the product formed. The output of the spectrophotometer can readily be coupled to a computer, chart recorder, or X-ray plotter or printer, and photometric methods are well suited to the continuous assay. But before you set up the continuous assay, you have to consider the following points. The choice of solution for setting up the zero absorbance. So for continuous system that produce a drop in absorbance, a solution containing relevant chromophore can also be used to set the zero absorbance if the instrument can read the negative values. Then we have the temperature control. So because the spectrophotometer will have the bulbs and other kinds of electronic instruments, electronic components, they will actually going to have the increasing temperature. So continuous photometric assay of enzyme can only be performed satisfactorily at a constant temperature. It is especially critical to ensure that the content of the cuvette are at a proper temperature. You know that the temperature is actually going to change the enzyme activity, number one, and the temperature can also actually be able to change the dissociation of that substrate, and temperature can actually be able to even make a lot of changes, right? So that's why that a constant temperature in the measuring cuvette is very important. And then starting the reactions, because time zero is very important. When you are actually setting up or when you are starting the reaction, that is very important. So it is critical to set the things so that the amount of enzyme added not result in such a quick reaction that a significant portion of it is over before even reading begins. If the rate of reaction is so fast that the mixing process must be rushed, the enzyme sample must be diluted to slow the reaction. So time zero is very important because you will going to say after time t, after time 10 minutes what will be the absorbance. But if you add like a large quantity of enzyme, if you add large quantity of, if you set up the reaction in such a way that it's going to complete before you go to that point or before you even start measuring, then it is actually going to give you more errors. Now let's see the examples of enzyme assays by the absorbance chains. So direct observation of reaction using the natural substrate, right? So a limited number of enzyme catalyzed reaction results in a usual change in absorbance. For example, the oxidation of NADH or the NADPH as well as the opposite reaction cause a significant change in absorbance at 340 nanometers because it has the epsilon 340 as 6200, and this simplifies the direct continuous absorbance assay of a large and important group of enzyme known as dehydrogenases. So for the dehydrogenases, what you can do is you can just add this NADH substrate and you can actually be able to monitor the disappearance of the NADH or reappearance of the molecules. So for example, you can actually be able to measure the lactate dehydrogenase. So lactate dehydrogenase is one of the most commonly measured enzyme because its presence in the serum after tissue damage aids in the clinical diagnosis. Because the reaction catalyte is freely reversible, the assay can be performed either in the direction of lactate oxidation by NAD plus or in the reverse direction by the pyruvate reduction by the NADH. Reduction of pyruvate, the preferred direction for the assay. For example, this, right? So pyruvate plus NAD plus NH plus gives you the lactate and NAD plus. So it is actually consuming the NADH, right? And you know that the NADH is actually going to absorb at 340 nanometer. So if you plot this, what will happen is that you are actually going to start, like for example, you start from 1, right? And over the course of time you will see that it is actually going down. So this is actually going to be like time versus product, right? So this is time versus substrate, right? So this is the NADH what you are measuring. So if you are actually going to see 10 molecules of NADH which is going to be consumed, this means the 10 molecules of pyruvate are also going to be consumed, and that's how there will be a 10 molecules of lactate which is going to be produced. And that's how you can use these informations to calculate the activity of lactate dehydrogenase. The enzyme may be satisfactorily assayed at 30 degrees Celsius, pH 7.2 in 50 millimolar trias with substrate concentration of 0.15 millimolar NADH and 1.2 millimolar sodium pyruvate. Then we have the indirect assay by coupling with the dehydrogenase. So because of the convenience of this change in absorbance when the nicotinamide coenzyme undergo oxidation or reduction, as well as the large number of dehydrogenases with a wide variety of specificities, these enzymes are frequently used as a final step in a coupled enzyme assays. So you can actually be able to couple these assays with the other enzyme, and that's how you can be able to measure the activity. Now let's talk about the another way of doing the enzyme assay. So the another enzyme assay way of doing the enzyme assay is turbidometry. So enzymes that act on insoluble polymers frequently clear the turbid solution, and this property can be used to calculate the amount of enzyme present. So turbidometry means the scattering of the light. Okay. So although the process is light scattering rather than absorbance, it can be measured with a standard spectrophotometer. Such turbidometric measurements are less easily standardized than the absorbance measurements, partly because the reproducible suspension of the insoluble polymeric substance are difficult to obtain, but are also for instrument reasons. Okay, so these are the places where you're not measuring the absorbance, but you are measuring the scattering. Okay, which means the scattering of the light and although the final result would be that you are going to see a disappearance of the intensity from the detector, and that's how you can actually be able to use this. But it has a limitation that it's not going to be very reproducible, because it depends on the how good your instrument is and how good your that scattering material is actually be remain in the solution, right? Some of the scattered light reaches the photodetector, and the proportion depends on the distance of the detector from the cuvette. So this is what is going to happen when you put the molecule in cuvette and it is having the scattering properties. It is actually going to scatter the light in all the directions, right? And some of the light is actually going to enter into the detector, which is actually going to affect the final results. So the enzyme lysozyme, for example, you can be able to hydrolyzes the bacterial cell wall and can be easily measured by observing the change in gravimetry that occur when it added to a suspension of dried bacterial cell. Because this decrease in turbidity is clear the result of a complex process of progressive random hydrolysis, the rate cannot be expressed in molar terms. So the unit is defined in terms of the rate of turbidity decrease. The wavelength for this turbidity measurement is arbitrary, set at 450 nanometer, and the one unit of activity is defined as the rate of change in absorbance that produced an initial rate change of 0.001 per minute, and when the volume in the cuvette is 2.6 ml. Okay. It is important to note that the volume in the cuvette must be specified in this particular case, because that is actually going to affect the settling process. When the same number of units are added to a smaller volume of this suspension, the turbidity change proportionally more. Then we have the third phenomena that is also can be used to measure the substrate or the product concentration is the fluorescence. So fluorescence like absorption is caused by an electronic transition that converts the absorbing molecule to an exciting state. As a result, the excitation and absorption are two words that describe the same physical process. What happened when the excitation state returned to the ground state distinguishes the fluorescent and non-fluorescent compound, right? So when you have a compound, for example, if you have the some compound, right, and if you illuminate this with a high beam of light, what will happen is that the electron in this is actually goes into a higher energy state, and then they come back to the energy. And in this process, and after that, they are actually going to give you a light emitting of light from these, right? So this lambda is going to be lambda 1, this is going to be lambda 2, and then lambda 2 is actually going to be bigger than lambda 1, because in this process there will be some loss of energy, and that's how the lambda 2 is going to be bigger than lambda 1. So whereas the energy of an exciting molecule is lost as heat in a non-fluorescent molecule, a fluorescent compound emits some of the energy as light, right? So during the 10 to the power minus 9 second interval between the absorption and emission, the molecule loses some of its energy through vibrational relaxation, resulting in a lower energy and as a result a higher wavelength than the exciting wavelength. Okay. So fluorescence can be used, and if you want to measure the fluorescence, you are going to have the different set of instrumentations. So this is a schematic diagram of a fluorimeter. So till this it is actually be same as the spectrophotometer. So you're going to have a light source, you're going to have the monochromators, and then you're going to have a slit, and then you're going to have a cuvette, and this cuvette is actually going to show you the light, right? Because it's going to show you the light in all the direction. And then since you in the spectrophotometer you have a detector on this side, so you can actually be able to measure the light what is actually being absorbed by this molecule. And but in this case what you're going to do is you're going to put another monochromator, so that you can be able to select the wavelength what you are actually going to see, and then you're going to have a slit, and then you're going to have a detector. Okay. So what you are actually basically doing is you are actually detecting the wavelength of the light for which, so this is actually going to be a lambda 1, this is going to be the lambda 2, and on a specific lambda 2 you are going to see whether that light is coming out from the sample or not. So the quantitation of the fluorescence. So although the condition can be set up so that the relationship between the measured fluorescent emission and the concentration is linear. The concentration of fluorophore in the sample cannot be calculated by applying a universal constant equivalent to absorbance coefficient to the measured the fluorescence emission. Okay, so fluorescence is not like absorbance, very straightforward that you can be able to just put the the molar absorption coefficient and you can actually be able to calculate the concentration of the compound. So a fluorescent compound emits a fixed percentage of the light it absorbs. That fraction is referred to as the quantum yield. And strong fluorescent material have the high absorption coefficient and a quantum yield close to 1, which means they absorb a lot of light and re-emit majority of it as a higher wavelength. Whereas in other cases, the quantum yield is going to be low, and that's how it is actually going to emit only a fraction of that absorbed light. Although quantum yield is constant for a given set of experimental conditions, several aspects of the instrument prevent it from being used for direct concentration measurement from fluorescence intensity measurements. The causes of non-linearity or the inner filter effect is that at the exciting wavelength, all the fluorescent compound absorb light. This means the light intensity of incandescent light decreases exponentially as the beam passes through a fluorescent compound solution. As a result, the relationship between the fluorescence emission and the fluorophore concentration is non-linear. This means I is equal to IOQF 1 minus 10 to the power ECL. As long as the absorbance value at both the exciting and emitting wavelength are sufficiently low, a non-linear relationship between concentration and fluorescence emission is possible. This means if you want to use the fluorescence as a assay to measure the substrate or the product, you always have to use the diluted samples. Examples of the fluorometric enzyme assays. So you can have the direct observation of the natural reactions. For example, the NADPH dependent glucose 6 phosphate dehydrogenase. So you remember that we have used the photometric assays also to measure the dehydrogenase assay. Same way, you can actually be able to use the fluorometric assays. So you can actually take the glucose 6 phosphate dehydrogenase by adding the 50 microliter of enzyme sample to a 3 ml of 0.1 molar trace pH 7.8 containing 3 millimolar of magnesium chloride, glucose 6 phosphate and 7 millimolar of NAD plus. And for NAD plus, the lambda excitation would be the 340 nanometer and lambda emission would be 465 nanometers. So this means you are going to excite it. Thus, you will keep the sample into the cuvette, then you will excite it at 340 nanometer, right? So in the monochromator 1, you are going to set as 340 nanometer, and in the monochromator 2, you are going to put as the 465. Then we have the phosphobilogen deaminase. So this is also another enzyme through which you can also be able to use to measure the enzyme activity. Then we have the ethylate synthase. So condition for the assay is given here, and ethylate synthase is catalyzing this reaction where the chorismate plus glutamate is getting converted into ethylenic acid and the pyruvate, and you can be able to measure this activity with the help of the fluorescence. Then sometimes we also use the synthetic fluorogenic substrate like the beta glucuronidase, chitinase, and elastase. So and many of the protease also are going to be used where you are using the fluorogenic protease substrate, and this when you are actually having the reactions, this fluorescence is actually going to be released. This fluorescent, probe is actually going to be released, and that's how it is actually going to give you the increase in fluorescence. Relief of the quenching. So an indigenous method for assaying the hydrolysis rely on a process known as the ratio-less energy transfer, in which from a fluorescent group on one part of polymer substance is transferred to a chromophore nearby in the same molecule without emitting the light. The process is interrupted when the substrate bond is hydrolyzed, separating the two interacting modes. Examples are carboxypeptidase, aminopeptidase, and phospholipase. Then we can also have the fluorogenic reagents like the amino oxidases, So using a continuous system in which the homoglinic acid is converted into a fluorophore in the presence of HRP, the enzyme that generates the hydrogen peroxide can be adaptive to a sensitive fluorometric assays. So diamine oxidase is assayed in a phosphate buffer containing homoglinic acid and 10 microgram per ml HRP. So this is HRP, right? Hydrogen peroxidase. Enzyme in the form of tissue sample is first mixed and shaken at 37 degrees Celsius for 10 minutes with buffer and the peroxidase before starting the reaction by adding the homoglinic acid and zero point 0.1 molar putrescine as substrate. The reaction is followed continuously at the lambda excitation of 315 nanometer and lambda emission as 425 nanometer. So, there are many ways in which you can be able to adopt the photometric assays, and that's why you can be able to measure the uh, the enzyme assays or enzyme activity. But as I said, you should be very, very careful when you are doing these kind of measurements, because it's a kind of a black box. What you are getting the absorbance, it has to be validated very carefully by running the control reactions and other kinds of things. So this is all about what we have discussed about the photometric assays, and in the photometric assays we have discussed about the UV visible spectroscopy, we discussed about the fluorescence spectroscopy, and we also discussed about the turbodistrict method. And all of these approaches are, are can be a potential way in which you can be able to use uh, to calculate the concentration of the substrate or the product, and that's how you can be able to determine the activity of the enzyme. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss more about the radiometric assays, and then we will discuss more about how you can be able to measure the activity in the gel itself, and so on. So with this, I would like to conclude my lecture here. Thank you.